Hi, I'm Adriana Goodwin, and today I'm going to be presenting a critical evaluation of HDAC8 inhibition via computer modeling. So some important background information for this project, just to understand the basics, is that a histone deacetylase, or an HDAC, is an enzyme that removes acetyl groups from histones. A histone is a protein that makes up a larger structure called a nucleosome, and a nucleosome is what DNA wraps around for it to be condensed into a chromosome. So because HDACs are removing acetyl groups, it's going to make DNA more inaccessible to genetic machinery. And this is a problem because HDACs are commonly overexpressed in many types of cancers, but mostly non-Hodgkin lymphomas, which account for about 20,000 of the 606,520 estimated cancer deaths in the US in 2020. And inhibition of this protein has the potential to increase growth, arrest, and apoptosis of tumor cells. So I chose to do a study specifically with HDAC8 because it is a class 1 HDAC, which means it is zinc dependent, and therefore the inhibitors can have different zinc binding groups that can interact with the zinc that's in the pocket of the HDAC. And if you look in the bottom left, you can see an image of the protein. And you can see that in the active site, it has a red zinc ion, and that is what's going to be bound by each of the inhibitors that were studied. And additionally, HDAC8s have been overexpressed in breast cancer specifically, which is why it is being studied in Dr. Ma's lab. So I chose to do an analysis of different inhibitors to see if there was a difference between zinc binding groups, and then also to look at the potential of the molecules that were developed in Dr. Ma's research lab. And in order to do this, I did a docking study, which basically was just determining the binding affinity and specific interactions between the protein and the inhibitors. So I chose to look at 10 inhibitors from the literature and 2 research molecules. And I completed the docking studies with modeling programs Gold and Mo. Gold was used for determining the interactions and scoring, and Mo was utilized for the final pictures. And as you can see in the center, I examined 3 different zinc binding groups: aminoamylide-based, hydroxamate-based, and cyclic tetrapeptide-based. Which is really just referring to the portion in the inhibitor that is binding the zinc ion of the HDAC8 protein. And in my chart, I've showed a range of chem scores, which again is just kind of looking at the binding affinity, and the higher the score, the better the inhibitor is at fitting into the pocket of HDAC8 and inhibiting its function. And I looked at the top scoring solution because I examined 30 conformations of the inhibitor fitting into the pocket. So if we take a closer look at the aminoamides, we can see that out of these two, A is the best inhibitor. And if we look at the docking images, we can see why. It conforms to the L shape of the pocket and has a lot of interactions with the protein residues that are less than 5 angstroms. And also, it has a lot of interaction with the zinc ion that's in the active site of the protein. Compared to the others I've modeled interactions less than 5 angstroms and interactions with the zinc ion. And you can just see that there's not as many, or it just doesn't fit in the pocket as well. And then if we move on, we look at the hydroxamate base, we can see that belinostat is the best hydroxamate based inhibitor. Again, if we look at the structures, we can see that it has that L shape, it has a lot of interaction with the zinc ion, and it has a lot of interaction with protein residues as well, compared to the others, which either don't fit in the pocket as well or have less interaction with protein residues. And then if we go back and we look at cyclic tetrapeptide, you can see that romidepsin is the only one that was studied here, and has a pretty low binding affinity, and that's likely due to the bulky structure not being able to fit into the L-shaped pocket. And if we look at the docking picture, we can see why. There's no interaction with the zinc ion, and there's not much interaction with protein residues. And then finally, we have gotten to the research molecules, and you can see that I modeled the top 4 solutions just to determine if there were any major differences in the conformations. And we're seeing that AOA115 and SCAO49 molecules have very similar chem scores to the aminoamylide and hydroxamate-based inhibitors of the literature. And again, I just modeled the interactions less than 5 angstroms with protein residues or the zinc ion. And out of these two, we see that AOA115 looks the most promising. But if we go back to the main poster, we can see that 2A overall had the best chem score and would likely be utilized for cancer treatments. And in the future, I plan on doing additional research from the literature on more characteristics such as HDAC selectivity, response rates, and synergistic activities to give a proper evaluation of which inhibitor would be the most promising for treatment.